This is a presentation demonstrating gene therapy with LCA and demonstrating the procedure of how gene therapy would be carried out in reality. Gene therapy is the procedure that could possibly fix a genetic problem at its source, and by adding a correct version of the defective gene, which would help the diseased organs and tissues of a living organism work correctly. Robert Johnson was the first person in the world to undergo the procedure of gene therapy for his LCA, which is Leber's congenital amaurosis, in 2007 in London, which was successful. Cells are taken from the eye and exposed to the AAV vector in a laboratory setting. So this isn't a laboratory setting, but let's pretend that it is. So this is my cells in my petri dish, and the eye have taken or removed from my eye, and this is the AAV vector, which is in the AAV vector is the adeno associative virus vector, and this is the red stuff, and so I'm going to expose it to the stuff, and this will not be accurate, but okay, so that is exposed, and vectors are used because injecting the corrected copy of the gene directly into the cell would not make the cell work properly, and so that's why you use a vector, because it injects itself into the cell and invades the cell. Also, viruses that are used to infect the cells are modified so that the person that is getting the genes injected into their eyes do not inherit the virus. Fluid from the eye is removed so that the RPE65 gene can be taken and put into the eye, and this gene is the process in which it processes a type of vitamin A, which to keep the light detecting photoreceptor cells, which are basically the cones and the rods in your eyes, and the genes are injected through a hair-thin needle, like so, and you want to put it into the eye, and you want to put it, and it goes underneath the retina, which is the red, and it goes underneath the retina to the cells which detect light, which are currently defective, and you want to insert the genes into the cells. You want to do this very carefully, because the retina is very, very, very delicate, and the retina could tear when you're doing the procedure, and you don't want that to happen, because that would fail the procedure, and that would be very, very bad. So, and also, when the gene has entered the cell and the information is traded back and forth between the cell and the genes, then protein molecules and RNA is produced. some disorders and not mental, medical conditions, excuse me, cannot be treated by gene therapy like others can. And for instance, genetic disorders, um, genetic disorders that are caused by mutations that involve one single gene are more likely to use gene therapy than, for instance, uh, genes that genetic disorders that involve multiple genes or multiple environmental factors, and that just shouldn't be used in that case because it's much too difficult and shouldn't be carried out. Side effects are not yet known because this procedure is very new and experimental, and it hasn't been used a whole lot. So long-term long-term side effects can develop years after the procedure is carried out, and short-term side effects are, for instance, you may experience headaches, fever, chills, severe chills, and the severe chills are also known as rigors, and also a drop in blood pressure, and vomiting, and a headache. And those resolve between or within 24 to 48 hours after the procedure, and these are caused by the immune system reacting to the vector that was injected into your cells, that invaded your cells. And so this concludes my presentation. I hope you enjoyed.